Kevin McCormack his subject is regenerating and reversing aging, a scientific approach. He likes short, pithy introductions, of which he has two, and I am taking the liberty of combining them both. Kevin is the communications director at California Institute for Regenerative Medicine, or CIRM, the state stem cell agency. The agency was created in 2004 by voters when they approved Proposition 71 and refunded in 2020 with $5.5 billion to fund stem cell research in California. Kevin is here to tell us about what we're doing with the money and how it's helped California become the global center of research. He is also the director of patient advocacy. He considers himself to be the unofficial or the official translator for the agency, working to turn complex language about equally complex science into everyday language that everyone can understand. Before joining the agency, he spent more than 20 years working as a journalist covering sports at the BBC before moving to San Francisco Bay Area to do health and medicine reporting for, I believe it was KRON. So discuss regenerating and reversing aging, a scientific approach. Please welcome Kevin McCormack.
Hello, everyone. Can you hear me okay?
Yeah.
Great. Thank you. Thank you, Patricia and Susan for inviting me. Patricia, very impressive chairpersonship. I like the way it moves along. Great stories, I have to say. I'm really impressed. Betty, I looked up chicken shit bingo. It's hilarious. And the one thing I do want is Ron Young's vocal cords. That's an amazing voice. I could make a fortune in voiceovers if I had your tonsils there on. So in keeping with everyone else telling jokes, I want to tell one that came first in the Edinburgh Fringe Festival, the one-liners. And it's man said, I went to a store. I was going to steal some spaghetti, but there was a female security guard and I couldn't get past her.
Oh.
I know, sorry. We'll put that behind us and hope we all forget that. And now we'll move on to the real business of me being here, which is me talking about stem cells. Can you see my slides? Excellent. So yeah, I'm with CIRM, with the State Stem Cell and Gene Therapy Agency, and we have a really simple mission, which is to accelerate stem cell treatments to patients with unmet medical needs. And this young boy is a perfect example of an unmet medical need. This is Ronnie Purwash, and he was born in Folsom, just up north of Sacramento. And at first, everything seemed fine. He looked really healthy and everything. And then he went home, but a newborn screening test found that he had a really rare condition called severe combined immunodeficiency, or SCID. Children born with this condition don't have a functioning immune system. So even a really simple infection can prove life-threatening. And children with this who are untreated often die within the first couple of years of life. It's also called bubble baby disease because children with this were something that used to be kept inside sterile plastic bubbles to help protect them from all sorts of diseases. So Ronnie was rushed to UCSF and he stayed there for the next 3 months. While they went through this new stem cell therapy that we were funding. And so basically what happened is that doctors Mort Cowan and Jennifer Puck took blood stem cells from Ronnie, and in the lab, they corrected the genetic mutation that causes SCID. They then grew those cells in the lab and then returned them to him. And because they're stem cells, they multiplied and grew and created more of themselves. And they created a new blood supply that was free of the genetic mutation, and that repaired Ronnie's immune system. This was Ronnie when he went home. He's now 5 years old. He started kindergarten. Actually, I went and had lunch with him and his parents a few weeks ago, and they were telling me that they'd been to India And this was Ronnie's first trip to India. So they checked with the doctor first to make sure it would be okay. They didn't want to do anything that might put him at risk. And they went to India. Ronnie was fine. His parents got sick, but Ronnie was fine. So clearly his immune system is doing well. We have funded research now that's helped cure 60 children of this rare disease. So that's quite a remarkable thing. The same approach is also being used to tackle another disease, sickle cell disease, and that affects around 100,000 people, mostly Black people in this country, but millions more all around the world. And again, they use that same approach. Well, they took, this is Evie, they took his stem cells, his blood stem cells, corrected the genetic mutation and returned them to him. Now, in the past, Evie had a lot of problems with pain, with crisis, with kind of organ damage and things like that. He was a really talented athlete at school. He had to stop playing football when he was in his teens because of the pain that was caused by this disease. It cost him a college scholarship. He couldn't really hold down a kind of a great job because he never knew when he was going to be in or out of the hospital. And often he was in the hospital for weeks at a time because of the severe pain that sickle cell disease causes. Now he's pretty much clear of the disease. This more than 2 years ago that he was treated. He hasn't had any sickling crisis since then. He's not taking any medications for this. He is now starting a new career. He's training to be a firefighter, which would never have been possible in the past. So it's really an encouraging sign for that. It's not just diseases of the blood, though, that we treat. We also treat strokes. This is Sonia Olia. Now, Sonia was in her early 30s, 32, I think, when she had a stroke. It was pretty devastating. It really kind of left her with almost no speech and really impaired movement on her right side. She couldn't raise her right arm above her shoulder. She couldn't really walk very far. Her leg was really weak. At the time, she was engaged to be married, but she told her fiance she didn't want to get married because she wouldn't be able to walk down the aisle. That had always been a dream of hers. But with such a kind of a withered leg, if you like, she said she would have to keep stopping. And she just couldn't imagine doing that. And so she said at the time, she thought her life as she knew it was pretty much over. And then she took part in a stem cell clinical trial at Stanford, not too far from here in the Bay Area. And what they did was basically take some donor stem cells from hematopoietic stem cells that had been kind of rejigged to be able to secrete more protective proteins for neurons and the other systems in the brain. And so those were transplanted into her skull near the site of the stroke. And to say that her recovery was fast would be a complete understatement, because the very next day, she woke up able to start speaking again in a way she hadn't for quite some time. She could raise her arm above her shoulder, and she could put weight on her leg much more than she had done. So over the course of several months, she went through some fairly intensive physical therapy to help rebuild the muscles that had kind of withered over the time. And she recovered really well. Now, this was amazing in two levels. One, because this is a phase 1 clinical trial. And in a phase 1 clinical trial, you're using a very small number of cells, less than is considered optimum, because it's the first time that's ever been tried in people. So you want to make sure that at the very least, you don't do more harm. So they use a kind of a very small number of cells. So to see that big an impact was really surprising. And it wasn't just with Sonia. There were 18 people in this trial. All of them benefited. There was even a woman who was 71 years old, and she benefited even more than Sonia, if you can imagine such a thing. So really impressive results. The other thing was that it used to be thought that maybe 6 to 18 months past your stroke, the level of recovery you had was pretty much the level of recovery you're going to get. You're not going to get much more, even with intensive physical therapy and everything else. Sonia was almost 3 years past her stroke. Many of the others were several years and up to 8 years past the stroke. So to see that level of recovery was really surprising. And it just shows that the brain is far more plastic than we imagined, that the ability to bounce back, to recover from serious traumas like a stroke is much greater than we ever believed. We're now funding a second trial with Gary Steinberg at Stanford, and we're kind of hopeful that that will prove even more impressive. So as for Sonia, her life now is almost back to normal. And I say almost because she's now married and she has a little boy, Lucian, who's just a darling little fella. So at this point, I want to kind of back up a bit and talk about what stem cells are, what they're not, what they can do and can't do, because I think there's still a lot of misunderstanding about this. So if you were to Google stem cell therapy, this is one of the first images that you'll get. It's shampoo and conditioner. And they're made from stem cells, but they're made from plant stem cells. So they're not really going to do you any good. And as proof of that, if they could help people, they'd help Boris, poor Boris. So what are stem cells? Well, basically they're you, they're me, they're all of us. Every single part of us began life as a stem cell. Our stem cells form every single part, from our hair on our head, our skin, all our organs, our bones. They all contain stem cells that help repair and replace the kind of things that get damaged with age and with disease. And now stem cells have a number of really important properties. Sorry, this is a cartoon I like to illustrate that point with, which is that sense of, just as the snowflakes make up every single part of the snowman and the snow woman, so stem cells make up every single part of us. And they have a number of really important properties. One is they can renew and replicate themselves over long periods of time. And for scientists and researchers, this is really important because you can take a relatively small number of cells and in the lab grow and multiply them into the millions and even billions of cells that you need to deliver a therapy. And the second quality is that they can turn into other kinds of cells in the body. And again, this is really important because you can take some fat, for example, and with simple manipulation, you can turn it into either bone or cartilage. You can take blood cells and turn them into every kind of blood cell, white cells, red cells, platelets, everything that you would need. And so at CIRM, what we're doing is we're funding the research to turn those cells into anything we want them to be, to turn them into the kind of cells found in the heart that get damaged in a heart attack, so we can repair the damage caused by a heart attack. The kind of cells found in the kidneys and liver, so you can help regenerate and repair those with diseases that have kind of ravaged them. And so far, we have funded more than 80, in fact, 81 clinical trials. We have another couple coming up this month that we probably will fund as well. And that's in everything from diabetes and heart disease, several different kinds of cancers, kidney disease, neurological disorders. So we're covering a broad range of topics here. We've also funded more than 24 other projects in early research where they eventually got into a clinical trial. And 3 of those have been approved by the FDA for use in patients. So that's really, really encouraging. And we're hoping over the next 10 years or more to be able to kind of increase that number dramatically and kind of help usher a lot more clinical therapies through the approval process. One of the best parts about being a state agency, where we get our funding from the state, is that we don't have to worry about making a profit. Particularly for rare diseases, that's important because there are a lot of rare diseases that affect relatively small numbers of people. Even something like sickle cell is 100,000 people. A research company, a therapeutics, a pharmaceutical company is going to look at that and think it's going to cost us $1 billion to develop a therapy and get it through all the approvals. We can't make a profit on that, considering how few people, relatively few people, have the disease. And at CIRM, we don't have to worry about that. We just look and see where the need is greatest and where the science is strongest. And we can fund that research in ways that no one else could. For example, the SCID, little Ronnie, that would never have been developed. In fact, there was a company in England that licensed that therapy. And because they realized after a while of testing that they couldn't make any money on it, they put it on the shelf. And this is a life-saving therapy, and they just put it on the shelf. Fortunately, we were able to work with UCLA and some others to pressure them into giving the license back to UCLA so that they could then, again, make it available to patients in need. So the beauty about being a state agency is that we can do those things. We have the power, we have the clout, we have the money, and we have the desire to help people with these really rare diseases. One of the things we're working on is not so rare, actually. It's age-related macular degeneration, and it's the leading cause of vision loss in the US in the elderly. And this lady, Anna Kuhl, is one of the examples of that. She lives in Marin, was really active, loved going out, doing things in nature, watching TV, movies with her husband. And then she got hit with age-related macular degeneration, and it came on quite quickly. So she went from being able to do things and living independently almost, and then to being almost legally blind. And it really stopped her doing any of the things that she really loved. Then she took part in a clinical trial that we funded with a company called Regenerative Patch Technologies. And the patch is that little thing that you see there. That's how tiny it is. And what it is a synthetic scaffold seeded with cells, embryonic stem cells that have been turned into these RPEs, the cells found at the back of the eye, the retina, that were being destroyed by this disease. And basically, they just inserted this to the back of the eye over the part of the retina that was being attacked by the disease. And over the course of several months, patients, there were, I think, 24 patients altogether in this trial, began to experience some benefits. 60% of these either experienced an improvement in their vision, or the disease stopped progressing. Because what they did was they implanted it in one eye, the eye with the worst vision, and they used the other eye as a control. So they were able to see if the patch was really working compared to how the other eye was. And in the untreated eye, the disease kept progressing. In the treated eye, in many cases, it didn't progress. And in some cases, it even reversed. Some of the patients developed kind of the vision improved. They were able to score much higher on the eye chart. Anna, for example, scored 17 letters better on the eye chart after the treatment than she did beforehand. And that was about 4 years ago. Her vision has continued to remain steady since then. And again, this company is working on developing a new clinical trial with even improved ways of delivering these therapies to the eye. So for Anna, now she's back doing the things she loved to do. She's able to go walking in nature, do many of the things that she hadn't been able to do in the past. And we're hoping with the advances in this treatment, we'll be able to kind of treat the other eye as well and hopefully restore some vision to that. And then finally, this is Byron Jenkins. He really is a top gun. That's Byron with his hand on the plane. And that's an F-14 Tomcat. Those are the planes that Tom Cruise flies in the movie Top Gun. And Byron was a Navy fighter pilot, and obviously in great shape, great physical shape. But a few years ago, he was at a birthday party. It was his daughter's birthday party, and she dared him to do a backflip into the swimming pool. And he did, and he almost died. His neck collapsed, his back was kind of broken. They had to kind of pull him out of the pool and rush him to the hospital. And it turned out he had multiple myeloma, which is a blood cancer that eats away at the bones. And that was the first notion that Byron had, that he had this. He lost 3 inches of height that day because his neck simply collapsed. And he spent the next 5 years in and out of the hospital, getting chemotherapy, surgery, radiation, blood transplants. Nothing really worked. And he said he was alive, but not really living. He had constant fatigue. He had no appetite. He said he was struggling just to keep going. And then he took part in a clinical trial that we funded with a company called Poseida that's actually just been bought up by Roche because they see the real potential in this therapy, where they took some of his own immune system cells. And the immune system is designed to protect us from infections like cancer and various other things. But obviously, in this case, the cancer had managed to evade the immune system and was attacking Byron. So in the lab, they re-educated these immune system cells so that they would recognize the cancer. They were able to identify the proteins on the cancer so that they could attack it. And then they returned those cells to Byron. And because they are stem cells, they grew and they multiplied. And Byron says that within 2 weeks, he felt so good, he stopped taking any medication. And he was able to leave the hospital. He stopped taking any medication, and he hasn't taken anything, even as strong as an aspirin, since then. That was 3 years ago. So he's now back with his family, doing all the things he wants to do. I don't know if he's doing backflips into the swimming pool, but he's going skiing, riding bikes, doing all the other things that he wanted to do. And that's thanks to the power of this therapy. And again, as I mentioned, Roche Pharmaceuticals has just bought the rights to this therapy because they see a tremendous potential to be able to reverse the course of a disease that in many cases is kind of fatal. And so this is pretty much who we are. Created in 2004. Refunded in 2020, got $5.5 billion to fund research. We've done over 1,000 different programs, from really basic research through to clinical trials, because we want to create a pipeline of products. We want to kind of reward the best science from the earliest stage, and then follow that all the way through and hopefully get it through approval from the FDA. 81 clinical trials and a number more coming along in the pipeline, and more than 3,400 patients that have been treated and enrolled. And so that's pretty much what I have to talk about, and I will happily take any questions.
That was absolutely eye-opening. If I may ask the first question, how does one get in a clinical trial?
It depends on the disease. Often if it's a rare disease and it's a phase 1 clinical trial, they're only going to be treating, say, 12 to 20 patients. So you have to go through your specialist. They can recommend you to it. Or there's a website called clinicaltrials.gov. And there you can search by different types of disease. You can add in things like stem cells or gene therapy. You can put a location in, not just say the US, but you can break it down by state and even city. So there are lots of options in terms of if you're looking for a clinical trial. And once you get to that page, it tells you here's the eligibility requirements and then the contact information. So there are lots of different ways. With the early stage stuff, though, it typically is done at, say, one of the major medical establishments or facilities in the state, say at UC San Diego or UCLA or UCSF and Stanford up here in the Bay Area. And so they'll often have a pool of patients who they think would be good for that particular trial. But there are lots of different ways. It just depends on the disease and the severity and everything else like that.
Anastasia has a question before I ask my second, being greedy and curious.
No, well, okay, we'll come back. Just seconding to that a little bit further. So I'm curious, okay, great news for people that might be in, near Stanford and UCSF and the other, okay, but
Yeah.
What about that person, where Randall's driving right now out in West Virginia, who might normally qualify? Is part of the funding to cover the expense of people being able to travel to said trials, or are they kind of out of luck?
Good question. We fund the clinical trials we fund are free to the patient, so they don't have to pay for the therapy. But right now, they often have to pay for transportation or travel to the clinical trial site. And they're only here in California that we do this. And or maybe even accommodation while they're getting the therapy. And so obviously that limits it for people who live in, say, rural parts of California or kind of in communities that aren't really well served. If you're, say, you have sickle cell disease, for example, many of these patients can't work because they can't hold down a job regularly because of the effect the disease has on them and the fact that they have to spend long periods of time in the hospital. So what we're trying to, and so they may not have a car. If they have a car, they may not be able to afford to park and stay with the kind of the high fees that many of these facilities charge. There's no bus service there, that kind of thing. So there are lots of obstacles. And so what we're working on now is we've created a patient assistance fund. And so when we make money on a therapy, or when a company makes money on a therapy, they have to return some of that money to us. We put that money in a patient assistance fund, and it will help, Anastasia, to cover those costs that you talk about, the cost of transportation to the facility, of staying at the facility, maybe even of paying for daycare for the kids or something like that. Because if you're a young family, and you don't have someone to look after your kids, what do you do with the kids while the mom or the dad is in the hospital getting this treatment? So there are lots of ways that we're trying to help that. We just started that. We've also created an accessibility and affordability working group so that we can make these therapies much more available to people all over California, not just in these major metropolitan areas. And the idea is that this group will be working not just in terms of this patient assistance fund to kind of put money aside for people who really can't afford to do that otherwise, but also working with insurance companies, working with state and federal governments to create a pathway so that when these therapies are approved, and they're going to be very expensive when they're approved, that they can be made available to the people of California, because we're helping pay for the development of the therapy. So it's only right that people here should have access to it. There was a treatment for beta thalassemia that was approved just the other week. It's $2 million. Now, in this case, it's a disease that is almost always fatal, and it has a long lifetime, and it will cost like $15 million to get treatment for that over the course of many years. So $2 million compared to $15 million, it's a bargain, but getting someone to put that money up is really difficult. So we're working on lots of different fronts to try and make these therapies both available and affordable to the people who need them.
Perfect. Thank you. And our next question comes from one of our guests. Now, I just want to, not sure if I'm pronouncing it right, Sakumalo? Unmute. Yes, perfect.
Hi, sorry, it's Sarah Kumalo. So
Good.
Thank you very much for allowing me to join, and also for an amazing presentation. Quick question, how does this therapy help an amputee, as an example? Is this something that you're looking at? Because you've talked about diseases, sickle cell, and so forth. Is there an opportunity to actually support any of the amputees, disabled individuals as well? Thank you.
Thank you, Sarah. Not at the moment. I mean, the stem cells are very much, the therapies that we're developing are very much for specific diseases. So in terms of amputees, it may be if there's nerve pain or nerve damage, it may be able to help them on that level. In terms of being able to regenerate, we're not there yet. There's a lot of research going on because we see in certain kind of animals, in certain kind of newts and salamanders, that they're able to regenerate a limb after it's been cut off. And so if we can figure out what's going on there, then obviously we might be able to help people with regeneration. I mean, that's very far down the road. I mean, people have the ability to do that. So for a newborn baby, for the first 3 months of life, if they lose their little finger, it will regrow back. But after 3 months, we lose that ability. So it's there somewhere. So it's a question of figuring out what's going on in the babies that for the first 3 months of life, they're able to do that. Secondly, can we then stimulate that and kind of recreate it for people later in life so they're able to regenerate limbs and things like that? But it's that kind of research is very much down the road, I'm afraid.
Alan Garber.
Hi, Kevin. Thank you for your presentation. In the first two examples that you gave with the newborn baby and stuff, you said that what they did is they took blood from the baby and then I guess extracted the stem cells, and then you said they did something to the stem cells to they changed them. What's that word that you used?
Yeah. So what they did is they take blood stem cells from the bone marrow. And the bone marrow is where it's all created. So, and in the lab, what they do is they delivered a corrected form of the gene to the cells.
So my question is, and this may be very technical, how do they do that?
They use what's called, there are a number of different ways. The one they used in this was called a lentiviral vector. It's basically a delivery system. You take a virus, and it can be something like the common cold, the flu, even HIV. You deactivate it so that it doesn't have any kind of infectious or disease carrying properties. And inside that, you insert the corrected form of the gene. You then use that virus to deliver the gene to the blood stem cells, where it kind of replaces the old one. And so then that becomes, you're able to generate these healthy cells. Another way of doing it is with something called CRISPR, which is a gene editing device. It's a bacteria that's found in nature, and what they do is they use that to kind of cut out the bad copy of the gene. And now they're developing ways where they use it not just to cut out the bad copy, but to replace it with a good copy of that gene, so that the person can then kind of develop healthy blood cells. And they're trying to use that for lots of other things as well. So CRISPR is going to be a really interesting tool that we're going to be using to repair all sorts of things in the future. Did that answer your question, Alan?
Yeah, it sounds like it's above the pay grade for most people in this room, though.
Mine too. I know the basic principle, but the mechanics, it's just, yeah.
Susan Rowan, would you like to state your comment to Kevin?
Well, first of all, thank you. You have proven how important science, experiment, encouraging our young people who are interested in STEM to be part of science. My own brother, as you all know, had polio, now has post-polio syndrome, and I happened to be in Chicago 3 days after he had a treatment. They took his own blood, whatever, put it into the CRISPR, the pot on the stove, I'm not sure. And I asked him how he was feeling, because they injected it into his left leg, which of course has been shriveled for years. And my brother said, this is the first time in decades I've been without pain. So to reinforce it. Now, the second part is my brother, who really is brilliant, except I said, oh, are you going back to have more? No, it takes too much time. Anyway, I don't know. How do we get him a lobotomy? No, question I have is, you talked about that this is something that we have voted on, this your organization. my great worry is with the amount of, oh, what would I call it? Crap that we are hearing about science. Are we sure that we can keep this organization that you're with operative if people have to vote when they're being fed a diet of baloney?
Mhm. Happily, California seems to be less prone to the kind of the skepticism about science than most other places. And with the passage of Prop 14 in 2020, it set aside this specific amount of money, $5.5 billion. So we don't need to go back to the voters every year or every couple of years to get refunded. We have this money, and this will last us, we estimate, between 10 and 15 years. So, and the hope is at that point, we'll have enough proof of concept that the work we're doing is really helping, is really benefiting, is proving so successful that people, if they feel like it, will be able to refund the agency again. It may be even that by then we'll have helped create the industry within California and made it so strong and so effective that we won't need an agency like ours to help kind of promote and stimulate this. But we're lucky in that we have, the money is set aside. So it's not as if the government can come in or the states can say, well, we're not going to do it this year. Other states have tried to copy what we've done, but instead of kind of setting aside a kind of a specific pot of money, they'll say, well, we'll fund you $50 million this year, 25 million this year, because things are a bit tough. That makes it hard to create a really effective and sustainable program, because if you're trying to build a laboratory, a lab with kind of all these postdocs and great researchers, you need to have a consistent funding source. And if you're worried from year to year that it's not going to be there, it's really hard to have that kind of consistency where you can work with a really basic idea and then progress it all the way through the different levels of research and take it into a company to be able to kind of make it available to patients. So we're very lucky here. And one of the things you talked about, Susan, where you talked about helping the young people. We have a number of education programs where we attempt to do just that, where we just had a high school internship over the summer where we take kids from different schools all around California, and they spend 8 weeks at a world-class research facility. Now, many of these kids are immigrants, first generation. Sorry if you can hear a lot of banging in the background. We got some people going on. The first generation immigrants. And so they're starting from scratch. They don't know much about stem cells. And when they go into these labs, I think they just talk about, well, I thought it'd be fun and I would be kind of rinsing out beakers and making coffee. But the day one they arrive, they're given a white lab coat, some goggles, and told this is going to be your project. And these kids are amazing. It's so much fun to see them at the end of it, where they deliver these posters of what the work that they've done. I have no idea what they're talking about, because it's just like they just soak up the science. And there are so many kids out there. And we have other programs as well for graduates and master's level students. So the idea is we're creating this program that's going to help people who may not otherwise be able to afford to do one of these programs, to get that experience, particularly from, say, low income or minority communities. This is a way of providing them with the funding that they need to help get through that. So we're creating this whole next generation of scientists, because we want to keep California the leader in this. We're a global leader. And if we can keep a continuous supply of kind of world-class scientists, then we've got a much better chance of maintaining that kind of leadership.
Perfect. Janice Litvin, Craig Adams, Carl Walsh, and then Betty. So Janice, you have a question? Is there a list of all the diseases you are now researching?
There's a list of all the work that we're funding. Yes, it's on our website, and that's www.CIRM.ca, California, .gov, G-O-V. So you go to that, and there's kind of a list of all the clinical trials we're funding, a list of all the other research projects we're funding. And you can break it down by disease or by kind of research type, whether it's really advanced research or more basic.
Craig Adams.
Kevin, thank you for your presentation today. I'm not sure if I enjoyed it more because of the content or it was just the level of eloquence you exhibited. I mean, it was really well done. You speak such gravitas. And so I just wanted to just shout you out for that, because it's taking a technical and scientific term and making it just understandable to the common man is such a skill, and you seem to have that ability. So
Thank you.
Thank you for displaying that.
No, I
Well,
mean, it's just, I don't have a science background. And so basically my approach is I ask the stupid questions so you don't have to. Like, what does that mean? What is this? When you talk about, so people talk about like oligodendrocyte progenitors, and I just like saying it, but those are cells in the central nervous system that are really important in helping repair spinal cord injuries. But if I went around talking about kind of transgenerational oligodendrocyte progenitors, people would go, what? And I wouldn't understand it either. So the idea is I try and kind of figure out what it is they're doing and explain it in a way that makes sense. And it's not dumbing down the science, but it's just making it understandable. And for me, I mean, that's part of what we should be doing. We're a state agency. We're getting funding from the people of California. So we should be able to explain what we're doing to the people of California in ways that they can understand and ways that they can appreciate how well their money is being used.
You are simplifying the complexity. As Shakespeare said, you think like a brilliant man and speak like the common man.
Very common in my case, I'm afraid.
Next, do you want to add your comment in person, Carl Walsh?
Certainly. The thing that concerns me is everybody probably remembers that around 20 years ago, there was a national movement to outlaw this kind of research that went all the way up to the White House. And are you seeing any kind of ruminations about that? And I would love to see some PR, a PR effort to let people know the successes that you're having so that they realize what this is, that this is for good and not to create a mutant race of human beings.
Yeah, I mean, one of the reasons why the agency was created in the first place was because of that very movement to crack down on the use of, back then it was embryonic stem cells, which at the time was pretty much the only game in town. And there were all these kind of ideas that we were taking babies and stealing cells from them or murdering babies to do this and that. We were going to be creating clones of people and all sorts of, as you say, mutant races. That was when President Bush, George W. Bush, put restrictions on the use of federal funding for research. That was, in a way, the kind of the driving force behind the creation of CIRM. What's happened since then, though, is that we found lots of other different kinds of stem cells to be able to work with. So we don't have to rely on embryonic stem cells anymore, although we still fund research in that because they're still considered the gold standard. We can use adult stem cells, such as like blood cells or donor cells from other people. We can use, there's even a new kind of cell called an induced pluripotent stem cell, an IPS. And what happens is that you can take a piece of skin from someone, and just by changing some factors in there, some genetic factors, switching some on and off, you can turn that back into an embryonic-like state and then turn it into any other kind of cell in the body. For example, you can turn it in a piece of skin from someone with Alzheimer's. You can turn it into a neuron, the kind of cell found in the brain that's affected by the disease. And even though it was a skin cell not so long ago, it now acts and behaves like a neuron. So you can begin to study in real time what's happening in the brain of someone with Alzheimer's. We've never been able to do that before, where you could, in the past, if you needed to see, the only way you could see it was with an autopsy after someone had died. And then the disease is so far advanced, you really don't, you see the after effects, but you don't understand the process. And so now we can look at what's going on in someone with early stage Alzheimer's and mid stage and late stage. We can begin to kind of parse out what's going on inside the brain. And even that notion, like all those tangles in there, the tau and everything that people, is that the cause or is that just a byproduct of what's going on? And so we're getting much closer to being able to use these cells in ways that we never could before. And so there's much less controversy now than there was. There has, I mean, during the President Trump's administration, they did crack down on the use of federal funds for fetal tissue research. And that really affected a number of projects. There was a big project at UCSF where they were using fetal tissue to try and develop a cure, a vaccine for HIV/AIDS. And that project just stopped because the restrictions were so severe. There was also a lot of talk about backing kind of banning funding for embryonic stem cell research. But that would be at the federal level. So the nice thing about CIRM is that we're a California state agency, so we're insulated from that. So whatever they do at the federal level doesn't really affect us here. In fact, in many ways, it encourages companies and researchers to move to California because it's much more stable here, much more consistent, and less dependent on the kind of the variations of who's in office in DC.
Betty Taish and then Bert Hill.
Kevin, thank you, Patricia. Kevin, thank you very much. And I concur with Craig with your ability to present such complex, challenging information at a very articulate and well understood level. So thank you. Having been a former nurse, this is a fascinating topic, and I really concur with the research. I know you mentioned the website to go to for a list of diseases, but offhand, do you know if there's anything for osteoarthritis for adults? Number two, childhood diabetes and rheumatoid arthritis. Do you know of any studies in that? Because those are such severe diseases for kids.
Yeah, there's a lot of research going on for both osteoarthritis and rheumatoid arthritis. Rheumatoid is harder because it's an immune system problem, and so trying to figure out what's going on there is challenging. Osteo is a little easier, and there's a lot more progress going on in that. Again, replacing some of the kind of the cartilage and everything else like that in arthritis is challenging because the cartilage has to be strong enough and robust enough to withstand all the daily pounding, but also soft enough to cushion things. So there's a lot of research going on, some clinical trials, but nothing we've seen yet that's been approved by the FDA. So, and when you say childhood diabetes, are you talking type 1 diabetes? Yeah, we've funded 4 clinical trials in that so far, and we're working with a company called ViaCyte, and they've developed a thing where you take, hang on a moment, it's like a credit card. Can you see this? And so basically it's about half the width of a credit card, but about as thick. And embryonic stem cells are seeded inside that have been turned into the to beta cells, the kind of cells found in the pancreas that should normally be producing insulin. But for whatever reason, because of the autoimmune disease, they're not doing that. And so the idea is that you can simply implant that under the skin in the back. And as blood flows over it, it can detect if blood sugar levels are too high or too low, and then regulate insulin. It can secrete insulin to help restore them to the levels that are healthy. And the hope is that that will eventually be able to help people avoid having to kind of take constant pinpricks of blood to measure the disease, to do insulin injections, that it will create a much more steady supply of insulin, much more consistent supply. So far, that's proving, again, it's challenging because it's an autoimmune disease. And when you're putting something into the body's initial reaction is to attack it. So we're trying to find ways around that doesn't require the people to use anti-suppressant medications. But ViaCyte was recently bought by another company because, again, that technology is so promising and so revolutionary in many ways that we're hopeful that in time they'll be able to do it. They've done some tests in patients, and it has been able to produce levels of insulin that we've never seen before. There are still objects and kind of obstacles in the way. Funding is always a big thing because it's such an expensive process to keep developing these machines, because the devices, to get the FDA to approve them, even being implanted in people, and then to kind of monitor them and see whether or not they're working. But we're funding a lot of research in that. There's a lot of research going on all around the US and Canada and in Europe as well, because diabetes, type 1 diabetes, is such a huge issue. And the hope obviously is that if it works for type 1 patients, then eventually it may be able to be used for type 2 patients who no longer are able to control their insulin production as well.
Wonderful, and I hope Sydney Mobell notices that Betty is also wearing her Sydney Mobell hourglass, which I think 35% of the women in attendance have spent money with Sydney Mobell. Good. Bert is next, but then Janice. I think you've had a couple of questions, and I think I breezed past you. So Bert, then Janice.
Yeah, there's a very good movie. It was shown on Nova and a number of award-winning movie called Human Nature that covers CRISPR. in a lot of detail. I know it well because my neighbor across the street produced the movie, and it actually has cameo of my two grandchildren in it. So it's a really good movie to explain CRISPR and how it works and how it developed. And so through PBS, I believe, I said it was on NOVA, but it may have been on public view also regularly. So I recommend it. Thank you.
Good. And if we had a reward or a prize for the best background, Bert Hill would not win it. Good. Janice Litvin, are you there?
I'm in my kitchen.
Which we hope we hope your kitchen will soon look better.
Yes. Yeah, we'll be moving back into it about a month from now.
All right, perfect. All right, Janice, are you there?
Yes, thank you so much. I love the presentation. I'm fascinated with all the science, though I was a math person, though I love the science. You don't know that I talk a lot about depression and burnout. And when you said the word neuron, my neurons started firing. So what the question that occurred to me was, when you said neuron, I started thinking about all the biochemical psychiatric problems, depression, anxiety, and all the other extreme conditions. Can you speak to?
Yeah, we actually, one of the conditions under Prop 14, where we got refunded with $5.5 billion, was that we devote at least $1.5 billion of that to diseases of the brain and the central nervous system, because those are obviously such huge issues. So things like Parkinson's, Alzheimer's, schizophrenia, mental health disorders. We have 3 mental health professionals, patient advocates on our board. So they really kind of keep a focus on that area to make sure that if there are opportunities to fund the research, that we take those. Diseases are like Alzheimer's and Parkinson's get a lot of attention already. Schizophrenia and some of the other bipolar disorders, things like that, they're really hard to understand what the cause is. So there's a lot of research at the basic level trying to figure out what's going on there. Because once we can figure out what's causing it, and then we can help develop therapies to help treat it. And the same with depression. There's so many kind of variants and so many kind of questions about what's going on that it's hard to kind of develop a treatment if you don't know what's behind it. But we're definitely funding research in that area. It's such a huge area of need. And so this $1.5 billion is going to go towards helping address some of those issues and hopefully coming up with some solutions as well.
Wonderful. My question is, sorry for interrupting, what is the major source of your funding?
It's from the state. It's sale of bonds.
Oh, bonds. What about private corporate donors?
We don't have any because we haven't needed any. And also it makes it kind of problematic if we take money from industry, because it then may help kind of influence the things that we do. What we do though is what we kind of de-risk the research. So we can invest say $5 million in a project and get it through to the point where it's ready to go into a clinical trial, fund that clinical trial. And then if it's showing really good results, then the big private pharmaceutical companies, investors, VC companies will come in and take over that project or invest in that project. We did a thing with researchers with Irv Weissman and his team at Stanford, where he had developed a therapy or an approach to treating cancer that was kind of chemotherapy and drug resistant. And he'd identified this marker on the cancer cells called a CD47. He called it the don't eat me marker. And so the idea is that cancer goes through and the immune system should attack it, but somehow this CD47 thing kind of acts as a cloak and says to the immune system, don't eat me, I'm all right. And so he developed an antibody that would enable us to uncloak that. And then so that then the immune system could attack the cancer, and the chemotherapy and all of those other agents could be used to attack not just the kind of the cancer cells, but cancer stem cells, which have an ability to evade chemotherapy, to kind of go dormant and then come back later, which is why cancers go into remission and then there's a relapse. So we funded that early stage research. Then we got him into clinical trials, and they created a company called 47 Inc. And that was bought by Gilead about 2 years ago for almost $5 billion. So the idea is we'll fund the initial research. get these companies and the researchers through what's called the valley of death, because it's really hard to get the funding for that, and then get them to the point where the companies want to invest in them, and then they'll have the deep pockets to take them the rest of the way. So that's we partner with industry in that sense. In fact, just yesterday we announced a new partnership called the Industry Resource Partnership, where companies that we're funding or researchers that we're funding and have access to Novo Nordisk and Bayer and some of these other big companies to the manufacturing process, to the seeds, stem cell socks. So the idea is that this can help accelerate research in ways that, because they need these facilities and we don't then have to pay for them. We can partner them with these companies. And the companies obviously will have then kind of a vested interest in seeing that these therapies progress and go as far as they can.
How do you prioritize what you're going to fund?
We don't. We had a big discussion when we were first started, like, do we say we're just going to focus on these diseases and really try and push that through? Or do we just say we'll fund it? We're agnostic in a way. We'll just take, if you have a really good idea, apply to us for funding. And we have an expert panel of researchers, all from outside of California, who review these applications and make recommendations about what we fund. And then those recommendations go to the board and the board votes to decide whether to fund them or not. So we have a fairly rigorous review process, and having sat in on some of these meetings, it's fascinating. They go into every single aspect of the research, including previous research that might support it or might contradict it. And so we have a really kind of thoughtful process, but it's also quite quick. If you apply to the NIH for funding, it can take a year. If you apply to us for funding, you can get your money from the day you applied, if you're successful, to 3 months. And so particularly if you're a company that wants to get into a clinical trial, that's the difference between success and failure. You need that money really fast. You can't afford to sit around because it's expensive. And the patients need those therapies as well. And so that's why we've created this really fast system that's both responsive to the kind of the best science, but also the greatest need.
And Mike Milstein came into our meeting late. And if Mike is there, your camera is off. But yes, Kevin would very much like to speak to your Kiwanis Club. And we have another guest, which is Tom's iPhone, which might in fact be my friend Tom Drews, who I invited. And Lauren Ward, would you like to unmute yourself and make your comment to Kevin? Because I know you're very enthusiastic.
My comment is that this is an inspiring presentation because it's restoring my faith in the wisdom of the California voters. Mm-hmm. It's an investment in the future, and we don't see much of that mentality, sadly, on the national scene. So there's great reasons for optimism listening to your presentation.
Thank you. Yeah, I mean, we've been very fortunate. I mean, no other state has done this. So to have the voters of California approve us not just once, but twice has been reassuring. I have to say, first time round, we got 59% of the vote. Second time round, we got 51%. So there was a kind of a nervous week after the election when we could see our lead kind of going beep, 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 beep. But as you only need 50+1 to win. So we got that. And so now we're back in business. And that's the best part, is that we're now kind of stable for the next 10 years. And hopefully in that time, we'll be able to make an even stronger case to the people of California that the faith in us was justified.
I do believe we have covered everyone who put a comment. If you would like to unmute and ask Kevin your question before I ask a concluding question. Yes, Alan Garber, we charge extra for a second question.
Okay, send me a bill. I'd rather owe it to you than cheat you out of it. Kevin, my question is about the board. Who's on the board and how are they appointed, or are they political appointees or what?
Yeah, so it's a 35-member board, which is pretty big. So, and they're appointed by the governor, lieutenant governor, state controller, treasurer. So there are very much appointees, but they're nominated. We can, or any group can nominate someone. So there are certain fixed positions like the chancellor of UCSF, UCLA, things like that, because those are the major academic research institutions that do a lot of this work. But we also have 10 12 patient advocates. We have several nurses, because obviously the nurses are the ones who are kind of in the forefront of all this, delivering the therapy. So we bring them on board because they bring a sensibility that maybe an academic researcher may not have. We have business interest in there because obviously business is a hugely important part of taking these ideas and commercializing them. So it's a really mixed board. Those four kind of state officers are the ones that kind of nominate or appoint them, but we all get to contribute to the thing and kind of recommend who they will have on the board.
So basically they're all picked by one political party in California.
No, they can be nominated by anyone. So
Okay.
Politically, we're kind of agnostic. We have a number of Republicans on the board. We have a number of people who are independents, and so it's not about politics at this point. I mean, they're all Democrats in government. So yes,
Right.
but we can nominate anyone we like.
Okay.
Yeah.
Thank you.
You're welcome.
Are there any other questions before I ask my concluding question of Kevin? Speak now or forever be quiet. All right. I must say, Jim Prost, every so often when the photograph of President Tony comes up, I do a double take thinking, is Tony with us? You are, well, Tony, our president, is walking across Spain, so he is with us in spirit. Kevin, you began your career in sports and you have now moved to medical research. Were you equally passionate about sports as you now obviously are about research?
Sports was like not working. I mean, sitting there watching Wimbledon, recording, like videoing and editing every single game, or watching soccer live at a time when you couldn't watch anything live in England, was just too much fun. And getting to write about it and see that. So that was fun. Medical it feels like more work, but even more fun on a different level, because you get to be able to see the face of medicine changing, the way we treat people, the way we treat diseases changing. And I get to meet these incredible researchers, some of the most amazing people in the world. And a lot of them are really just, not just super smart, but also really nice and kind and humble. And some are raging egomaniacs, but that's okay. As long as they come up with a cure for cancer, we can put up with that. But I also get to meet the patients and become friends with them. These are pioneers. I mean, these are people who are volunteering for experimental therapies. They're putting their life on the line. And in many cases, they know it may not necessarily benefit them. But they're willing to do that because they feel that maybe the knowledge gained, the scientific data gained, can help inform something that develops a therapy that's going to help many more people down the road. So yeah, I mean, it's an amazing, I really feel privileged to have this kind of job where I can meet these people and work with these people on a daily basis. So yeah, but I still support Spurs in London. So that's a good thing.
All right, good. And Kevin, is this your third visit to the Breakfast Club? I know, was it twice in person?
Twice in person, yes. And now virtual. Maybe the next time I come back around, it'll be back in person again. I hope so. It was always a fun place to have breakfast.
Well, we certainly, whether it's virtual or in person, we hope we can invite you back in another year. And I look forward to our walk on the 3rd of September.
Indeed.
And Susan, Roanne, and I are especially pleased because Kevin married one of our very good friends. So he is part of our extended family. Susan?
Yeah, and Kevin has been active in science and medicine because when he was at KRON, when it was our NBC affiliate, Kevin and I spoke because I was one of the people that started the Red Hot Mamas, the women who talked about menopause before anyone else did in the 1990s. And Kevin interviewed me. So Kevin has been active in pursuing science and cures and anything that helps people. So I go back with Kevin, I think even before you met Shirley. So but he wanted to bring to the public what we women knew about taking care of menopause. So thank you, dear.
No, thank you. I always remember, like, who am I going to get to talk about this? Because no one talked about it. And then I found this group, and then Susan said, yeah, ask me any questions.
May I say one thing, Patricia?
Derek, yes.
Kevin, I thought you were fantastic, brilliant presentation until you said you supported Spurs. Now, I'm afraid you've gone down in total credibility because I'm a Gooner, and you probably know what that is. An Arsenal.
Yeah.
For those that don't know, they're a mile apart, and there's a lot of bad blood between the teams. But
sadly, stem cells can cure many things, but not you, Derek. I'm afraid that's a terminal condition.
Now, Derek, on your video chat show on a Monday in England, would your audience be interested in hearing what we do in California?
I think they'd be very interested in some of it. It may go over our heads, and Kevin would understand that as a BBC journalist. But I'd love to interview Kevin with you, Patricia, and maybe we could talk about your time in London, what you learned from supporting Spurs, and
tolerance, patience, and a great ability to kind of put up with pain and disappointment.
Good. Well, I will introduce you after this meeting, and thank you. Let's give Kevin his applause, and then I'm going to ask Pete Ratto to give us a preview on next week.
Thank you, everyone. It was great. Great questions.
Thank you, Kevin, and we look forward to seeing you perhaps next year, and some of us like me will see you earlier.
Yes,
so
indeed.
Thank you, wonderful, and your background is great. It really looks very impressive, just the way a man who is dedicated to science would have his real office look like. So with that, Pete Ratto, would you like to give us a promo on your program for next